So welcome back, and I'm very happy to be moderating this next session on a whim, which is wonderful. But while we're just resolving or getting over this technology hurdle here, I thought I'd take a moment to hijack a couple of minutes here. It is Mitochondrial Disease Awareness Week, which is a global initiative, funnily enough, celebrated around the world. There's a tautology if ever I've had one. And as part of Global Mitochondrial Disease Awareness Week, for the first time this year, the Australian Mitochondrial Disease Foundation called for applications and for submissions and recommendations for researchers to submit a publication that they'd attained related to mitochondrial disease in the last financial year, so from 1st of July 2014 up to 30 June 2015. And because the members of the AMDF's Scientific Medical Advisory Panel seem to know pretty much everybody in the mitochondrial disease research field in Australia, and we're inherently conflicted, we outsourced the review of those applications to our sister organisation in the United States, the United Mitochondrial Disease Foundation, who generously offered the services of their advisory board to review the applications, and that was all done over the past couple of weeks in anticipation of Mitochondrial Disease Awareness Week. And we're pleased to we were pleased to announce 2 awards for excellence in mitochondrial disease research. The first was awarded, there were 2, they're in no particular order. We'll go in reverse alphabetical order. The first one was Dr. Manal Menezes, who received her award in person at our presentation on Tuesday in Sydney. And I'm very happy to invite Luke Formosa, who happens to be here today, up. And we'd like to present you, Luke, with our second in reverse alphabetical order award for excellence in mitochondrial disease research. And there's a little an incentive as part of that, a travel scholarship for you to use as in your next or future research-related travel pursuits. So would you join me in congratulating Luke for winning this award? And you don't get to speak. Thank you. So we're about to go into the second session. We've got 4 presentations that are very diverse in nature and really starting to explore a lot of the other things that hang around something like mitochondrial donation, from some more of the science to the legal and ethical considerations. And then we're also going to hear about what happened in the UK. How did this all come to be, and where is it at the moment? And we're going to finish off with a panel discussion. And so I'm not going to allow questions until then. You can hold them back, but please hold them back. And we'll get everybody up for hopefully a challenging and robust panel discussion at the end. So our first speaker in the second session is Dr. Damian Dowling from Monash University. He's the Senior Lecturer in the School of Biological Sciences. And Damian's going to talk to us today about some of the considerations there around mitochondrial donation and some of the findings of some of his and some of his peers' research around mitochondrial donation. So please welcome Damian Dowling.
Many thanks, Sean. It's a pleasure to be here today, and thank you also to John, who invited me to come today to give this presentation and outline a case for caution when it comes to mitochondrial donation, if indeed there is one. It's obviously a highly promising technology, and many, if not all of us in the room today, would be supportive of it in principle, given that it offers hope to females of reproductive age who carry pathogenic mtDNA mutations to have their own biological offspring that might or should be free of carrying mtDNA-mediated mitochondrial disease. So it's a highly promising technology. It comes with a couple of issues that should still be on the radar of people involved in pushing this technique out and legislating it. These issues we've heard a bit about today already, quite extensively, the issue of carryover. So carryover of mutated mtDNA from the mothers into the offspring and the resulting heteroplasmy that creates. And the other issue which Mary talked about briefly, was that mitochondrial donation creates novel combinations of mitochondrial and nuclear genotypes. And that has the potential, at least in theory, okay, according to robust evolutionary and population genetic theory, to result in incompatibilities between the mitochondrial and the nuclear genome. So the efficiency of the technique is excellent, but low rates of carryover of mother's mutant mtDNA are still likely following donation. And we've seen these slides today. This one's taken from a recent paper in Trends in Molecular Medicine, and it shows the spindle transfer technique, whereby the patient's egg that contains the mutant mtDNA, the spindle is isolated into a carrioplast. It's then translocated, transplanted into a enucleated donor egg, and that will then be fertilized to create the functional zygote, which contains the patient mother's nuclear chromosomes, so inside the carrioplast, and the donated mitochondria, complete with the donated mtDNA. So as we now, as it's clear, there is some carryover of the mother's mtDNA. And this study from Lee and colleagues from Oregon, published in 2012, Cell Reports, reports from the macaque model that the copy number in the carrioplast is about 0.6%. So this equates to about 3,500 mtDNA molecules from the mother, which are then placed alongside the donor's 500,000 or so mtDNA molecules. So carryover is very low. We're talking about less than 2%. And we now know that the critical threshold by which mutant mtDNA molecules need to increase within a given tissue to cause mitochondrial disease needs to be typically in the order of about 80 to 90%. So what's the big issue? The issue is the fact that the levels of mother's mtDNA that are found in the, let's say, fertilized zygote or in the offspring when they're just born, not necessarily going to hold constant throughout the life of those offspring. So here we have 4 macaques born post spindle transfer. This is research out of Oregon. And you can see that in 3 out of the 4 live births that were produced following mitochondrial donation, 3 of the 4 had carried over levels of their mother's mtDNA in their cord blood at birth. 1.5 years later, they still possess this carried over mtDNA. Generally, these levels had changed. In one case, they had gone down. In a couple, they had gone up. So it's not so much of a problem here unless there is a directional trend for carried over mtDNA to increase throughout the life of the offspring. And this can be, as has been discussed by Mary and David, via the bottleneck, and it can happen randomly. The concern would be if it happens non-randomly via a selfish drive that is associated with mutant mtDNA molecules. So here's a little bit more research from these macaques showing that the embryos produced following mitochondrial donation via spindle transfer initially had less than about 0.6% mtDNA carryover throughout the somatic organs. But when they looked at the oocytes in 2 of the fetuses from singleton births, they found that about 8% of these oocytes had quite high levels of mtDNA carryover. Right? So we're talking more than 10%. This could, of course, be due to the bottleneck, or it could be due to preferential replication. So what sort of evidence do we have for a selfish drive associated with different mtDNA haplotypes, or even more specifically associated with functionally impaired mtDNA molecules? There is quite a bit accruing across the eukaryote kingdom. The most relevant comes from this study in 1992 by Yanida and colleagues showing a marked replicative advantage of human mtDNA carrying the mutation that causes the MELAS syndrome. But we also have many examples in the animal kingdom. So we know about the petite mutations in yeast, which are functionally impaired but very good replicators. The same is the case in fungi. When we look at these closer to our own species, but we're still very diverged. Obviously, we're looking at nematodes here. There are functionally impaired mtDNA molecules that segregate in wild populations that contain a deletion. It takes out a little bit of the ND5 gene. Individuals suffer if they carry this mtDNA haplotype, but they're very good replicators, and therefore they increase in frequency within the population. Evidence for preferential selection of different mtDNA haplotypes when they're placed together in heteroplasmy in mice. This study comes from Brendan Battersby and Eric Shoebridge, and they looked at patterns of tissue-dependent selection. This particular example came from cells descended from the liver. And when they placed two putatively healthy mtDNA haplotypes inside the same mice, they found that there was a strong bias for one of these mtDNA haplotypes to increase in frequency over a 1.5-year period. In this case, it was the NZB mtDNA haplotype, which was outcompeting the mtDNA haplotype that had been sampled from the bulb strain of mice. So there's some chance that the patient's mutated mtDNA, although initially present at very low frequencies within the offspring, will preferentially replicate toward the level of the critical energy threshold. So it's something that we should keep our eye out, and it should be on our radar as this technique progresses through to the clinic. So the next issue that I want to talk about, and the final issue I'll talk about today, is the issue of mitonuclear incompatibilities. Energy production hinges on coordinated interactions between genes that span two genomes, mitochondrial and nuclear. It's very plausible to suggest that complex life forms and the evolution of complex eukaryotes is only made possible via these interactions between mitochondrial and nuclear genotype. Okay? This has allowed us to harness a very efficient form of ATP production. So these genomes co-evolve. Yep. There's no doubt about that. They co-evolve. Now, mitochondrial donation does something that sexual conception or standard IVF cannot achieve, because it has the potential to create uniquely novel combinations of mtDNA haplotype and nuclear genotype. So I'm going to give a little crash course in mitonuclear genetics, because I feel that this issue has been poorly represented in the biomedical literature when it comes to handling this potential safety issue. So here we have a female. It's a very simple schematic representation of a female karyotype. Her mtDNA is indicated by this solid red circle. Her nuclear genes, and in this example she has only 3 nuclear genes, gene 1, 2 and 3, are present in diploid form. Okay, she's got 2 copies, just like us, but we have more than 3 genes. Now, at oogenesis, the female will produce eggs of the following genotypes, again in haploid form now. Okay? And if she goes on to reproduce with a male, and these eggs are eventually fertilized, the zygotes will have the following genotypes, in which the male now contributes a whole haploid copy of his nuclear genome. He has different nuclear genetic variants. Okay? So we now have heterozygous expression of each gene. The key point I want to convey here, and this is essential for understanding of this issue, is that invariably 50% of the mother's nuclear genes co-transmit with her mtDNA every single generation, perpetually throughout evolutionary history. So let's now assume that a mother carries a pathogenic mtDNA mutation whose effects are offset by a modifier allele that lies within the nuclear genome. So this is a hypothetical situation. Under this assumption, we're going to assume that this particular mitonuclear epistatic allelic pairing, so a pairing of alleles, is essential for survival of the zygote. So the same simple karyotype, but this time we have our mtDNA mutation located within the mtDNA of the mother, and we have our nuclear modifier allele located on gene 1. Okay, in this case, it's present in homozygous form, okay, on each copy of the gene. So when she produces the eggs of the following genotypes, you can see that each of these eggs has the mtDNA mutation, but the phenotype is rescued by the modifier allele that has been co-inherited, okay, co-transmitted in every single case. Doesn't matter what's happening with the dad, we're only looking at the mother's genotype. They're all okay. Selection, natural selection, gives it the thumbs up tick of approval. That's nature's quality control screening process. She then mates with a male. Again, we see these same genotypes created. Half of these zygotes will turn out to be males on average, half will turn out to be females on average, right? Now let's take one of these females of this genotype and see what happens when she gets to meiosis, okay, and when she ultimately reproduces as an adult. Okay, we have this simple genotype. She produces eggs of the following genotypes, and now you can see that not all of these oocytes carry the nuclear modifier allele that's necessary to restore fitness to the zygote. So what might happen during, for instance, follicular atresia is that these genotypes that don't carry the essential mitonuclear pairing simply do not develop into secondary oocytes. Or if selection isn't strong or isn't manifested at that stage, they might end up being fertilized by a male when she's of reproductive age, and they're the genotypes you see. The male never passes his mtDNA on to his offspring, but he passes a haploid copy of his nuclear genome. But this is essentially irrelevant. Okay? There is no dilution in the face of natural selection. It's not just about Mendel's laws of segregation. We, complex life form, presumably could not be here without very strong forces of natural selection shaping our evolution. Okay, so selection should purge these eggs and let these ones through. If that's under the assumption that mitonuclear compatibility is important. And if it is, then natural selection will simply preserve essential mitonuclear gene pairings. This might happen during follicular atresia or prior to implementation of the fertilized zygote. And if this happens, a pregnancy will never have even been observed. Or it might happen in later life stages. In theory, preservation of co-evolved mitonuclear allelic pairings relies critically on the co-transmission of 50% of the mother's nuclear DNA with her mtDNA. That's the essential point of those slides. Co-transmission. Under mitochondrial donation, co-transmission between a mother's mtDNA and her nuclear genes is no longer 50%, it's 0%. This takes away the mechanism by which natural selection can preserve essential mitonuclear allelic combinations. The implication is that if co-evolved mitonuclear complexes exist and are important, then under mitochondrial donation, the rate at which mitonuclear incompatibility should manifest will be higher than under standard sexual conception. So that's the theory. What's happening? Well, there have been mitochondrial donation techniques, practice, or techniques that are analogous. They do the same thing as mitochondrial donation in a range of different model species, either when the nuclear backgrounds were inbred or when the nuclear backgrounds were outbred. And what we see in all of these examples, in mice, in Drosophila fruit flies, in other arthropod species, is altered phenotypes post-mitochondrial donation. Not necessarily altered for the worse, just altered. In many cases, altered for the worse though. So things like reduced cognition in mice following mitochondrial donation, reduced fertility in males, impaired male but not female mitochondrial function, altered survival, reduced mitochondrial function and ATP production. Well, that's the model species. Are nuclear modifiers important to humans? There is a wealth of evidence to say, yes, nuclear modifiers are important. Some of this research comes out of human cell lines, some of it comes out of association studies. Okay? We know when it comes to mtDNA-mediated disease that causes hearing loss. we know that there are nuclear modifiers that rescue that phenotype. Okay? Many studies, okay? A nuclear modifier for a mitochondrial DNA disorder in humans. Okay? It's at the 12S rRNA gene. Similarly, when it comes to mitochondrial mtDNA-mediated cardiomyopathy, we know there's a nuclear modifier that rescues the phenotype. And, as you heard earlier today, there are diseases which are traditionally not called mitochondrial diseases, things like Parkinson's, some cancers, multiple sclerosis. We know that these diseases now are mitigated. Their penetrance is mitigated by mitonuclear interactions. And this came out last week, the Kalman syndrome phenotype, which impairs development to puberty. Mitonuclear interactions synergize to produce the Kalman syndrome phenotype. Many more examples from the human literature, with this conclusion that disrupting mitonuclear coevolution affects OXPHOS complex 1 integrity and impacts human health. Mitochondrial donation is a highly promising technique, and it offers hope to sufferers of mitochondrial disease. There are some potential issues pertaining to health implications in the resulting offspring that we need to keep an eye on as this technique progresses to the clinic. I don't think it's good enough to say these issues are hypothetical in nature, because they're substantiated by experimental evidence, not only from model species, but genomic evidence in humans. So we shouldn't just sweep that evidence under the carpet. We should talk about it. It should be on the radar as we progress this to the clinic. Okay, thank you.
Thanks very much, Damian. You did very well in terms of time. So if there are one or two burning questions, we've got a minute or two now for Damian, if anybody has that. Mary, please speak up.
So the maternal genome is composed of quality function and lack of function of the genome. So what is co-evolution on the maternal genome, the genes she got from that, they wouldn't co-evolve in this specific generation?
No, they just, yes, unless, of course, they were better in terms of their synergistic interaction with the mitochondrial genome of the mother. But all that's required for natural selection to do its job is some level of co-transmission between the mother's nuclear DNA and her mtDNA. And in the case of X chromosomes, that level of co-transmission is actually very high, 67%, right? Because the X chromosome spends 2/3 of its time with females. Males only carry one copy of the X. So that's why we expect that strong mitonuclear adaptation is driven by interactions between mtDNA genes and X-linked genes. Right? And I think we get that with levers optical hereditary. There seems to be nuclear modifiers that seem that due to the X-linked.
So, for example, chromosome 11, for example, which includes a lot of genes that are important for mitochondrial function. Is there any evidence that the maternal grandmother's chromosomes are preferentially passed on? So we would expect in our population that if this coevolution theory is correct, that there would be a predominance of, for example, chromosome 11 from the maternal grandmother. Is there any evidence for that?
Yes, so a paper came out last week in Proceedings of the Royal Society B, investigating looking at whole human genomes across, I think, 100 different populations. They were looking for linkage disequilibrium between mtDNA haplotypes, so it's a whole haplotypes, and allelic variation segregating in the nuclear genome. And they found evidence for statistically significant evidence for mitonuclear linkage disequilibrium. Where particularly it was strong on the X chromosome, like I just argued, which is what I would predict based on levels of co-transmission between mtDNA and X. So where do the nuclear modifiers have to lie? Do they have to be nuclear encoded mitochondrial genes? No, they don't. And some of these modifiers I'm showing you, they're not. Indeed, in our fruit fly studies, when we place different mitochondrial haplotypes alongside isogenic nuclear backgrounds, we see that 10% of the male genome just goes haywire in its gene expression when placed alongside different mitochondrial haplotypes. They are nuclear genes that are highly sensitive to the mitochondrial genome they're placed alongside. Most of them are not most of those genes don't have mitochondrial annotations. Okay? They're in fact important in male they're overexpressed in the male reproductive tissues and things like that.
I tell you what we might do, thank you, is David, did you want to ask? I think we might continue. I think there's going to be a lot more discussions around that in the panel discussion. So we might please thank Damian for his presentation.